Okay, we are going to talk about Chapter 1 of this course, Chem 375, which is a survey course of all of the quarters of biochemistry put into one course. So we'll cover a lot of topics in a little less depth than we would if this was a majors biochemistry class. So today what I'm going to do, what Chapter 1 does is it really just is a big overview of things that we're going to cover in a lot more detail in the next few chapters. So I'm just going to take some time to go over some of the most important points before we get into Chapter 2, which is all about water. So let's talk about water first. So we are going to focus on some of the important features of water that make it special and conducive to life. We'll talk about water's polarity. We'll talk about its Lewis structure. We'll talk about its electron pair geometry and its molecular geometry and some of the things that make water as a molecule special, and some parts and properties of water that are required for living things. One of the most important properties of water that we're going to find to be very important in biochemistry and how chemical reactions happen in our cells is hydrogen bonding. So you will remember that hydrogen bonding, the name a hydrogen bond, is actually a very misleading term because a hydrogen bond is not a bond. It's a strong attraction. It's a special example of a dipole-dipole interaction between extremely electronegative elements. So a hydrogen bond is between a hydrogen that is covalently bonded to an oxygen, nitrogen, or fluorine. And that hydrogen can then be attracted to either another oxygen, nitrogen, or fluorine. So the thing that is important, and these are three different situations, and it is not true that the hydrogen is covalently bonded to an oxygen and then it has to be hydrogen bonded to another oxygen. You could exchange or replace this oxygen, nitrogen, or fluorine with each other. I just drew it this way, but we'll see a lot. We won't see any examples in biochemistry of fluorine in our cells, hydrogen bonds between fluorine. We will see a lot of hydrogen bonding between hydrogens that are covalently bonded to oxygens and nitrogens and then are attracted to other oxygens or nitrogens. So this solid line here represents a covalent bond. And then this dashed line here, this represents the hydrogen bond, which we already said is not a bond. It's an attraction, which is why it is usually represented with a dashed line, and an actual bond is represented with a solid line. So we'll spend a lot of time talking about this because a lot of the ways that the molecules inside our cells act has to do with hydrogen bonding and what if water is present or not. So we'll spend a lot of time talking about water and all of this. Another topic that we'll spend a lot of time talking about is thermodynamics. Thermodynamics in living cells. We mostly will be talking about the flow of energy. And the flow of energy in cells, we'll be looking at all of the chemical reactions in a cell, and this word is called metabolism. Metabolism really represents the sum of all the chemical reactions. For us, that will be in a cell. We'll talk a little bit about types of cells in this class. Our cells are called eukaryotic cells. There's prokaryotic and eukaryotic. We will mostly be talking about processes in eukaryotic cells. These are our cells. Prokaryotic cells are mostly things like bacteria. Prokaryotic cells have no membrane bound organelles. Eukaryotic cells do have membrane-bound organelles. The one thing that differentiates a prokaryotic cell, well, there are a few, but one of them is that it has a membrane-bound nucleus. U means true. Karyote usually refers to the genetic material. So a eukaryotic cell has a true nucleus, meaning a membrane-bound nucleus. Most of the other organelles in a eukaryotic cell are also membrane-bound, but this is really the one thing that qualifies a eukaryotic cell, is that it is membrane-bound. And these are what our cells are. We will talk a little bit about prokaryotic cells when we talk about how different processes were discovered. Usually we discover processes or isolate types of proteins in a more simple model, like E. coli, for example, is the model that you'll see used frequently. E. coli is a prokaryotic cell. So what we know about how things work in E. coli, we can then apply to eukaryotic cells that are much more complicated and then kind of see the similarities and differences. So if we're looking at pictures of enzymes or proteins and it might say that this was isolated from E. coli, that means it was isolated from a prokaryotic cell, and then we can apply that to what we know about eukaryotic cells. So we'll talk about this kind of more later. Some of you might have a strong biology background, and this will be a lot of review. Some of you might not, and I will try to provide a lot of explanation for people that don't come from a strong biology background. Okay, so back to thermodynamics. All of the chemical reactions in our cell, the sum of all of those is called metabolism. So there's two types of metabolic reactions. There are catabolic and anabolic reactions. Let's talk about catabolic reactions for a minute. So catabolic reactions are reactions that break things apart. So we'd be going from a bigger molecule to a smaller molecule. We're breaking things apart. Catabolic reactions release ATP, or you could say produce ATP. ATP is the energy, one of the energy molecules that we use, by far the most common that we will talk about, ATP. These reactions are called exergonic. Catabolic reactions, they release energy. We'll spend a lot of time talking about metabolism and catabolic reactions. We'll spend less time talking about anabolic reactions, where we're building things. We're building larger molecules from smaller molecules. So we might take two smaller molecules and add them together to form a larger molecule. This would be an anabolic process. Anabolic processes require ATP, require energy, and we call these endergonic. So this is how metabolism works. We have reactions that produce energy, and then we have reactions that require energy. And most of the time we couple these processes to get work done inside our cells. So we'll spend a lot of time talking about metabolism, talking about how we use the chemical bonds in our food to break those molecules of our food apart to produce energy. And then that energy that we use from the breaking of the bonds of the molecules in our food, we will use for all of the anabolic processes in our cells, to build proteins, to do mechanical work inside our cells, that kind of stuff. So we'll spend a lot of time on this, and this is all a type of thermodynamics. The last thing that we're going to talk about are the classes of macromolecules. And we'll talk about macromolecules throughout all of this. There are 4 classes of macromolecules. 3 of the 4 classes can be used for energy. The one that cannot be used for energy is nucleic acids. And those are used to record information, or it's an energy transduction molecule like ATP. The other 3 we use for energy. You may have heard of a diet called the macros, counting macromolecules. So you're literally seeing how much carbohydrates, fats, and proteins you're eating. So those are the macros, are these macromolecules. We don't include nucleic acids in that because, again, they do not provide energy. So let's start with each of these. We're going to name the macromolecule. We're going to talk about what its monomer form is called. Mono means one. We'll talk about what its polymer form is. Poly means more than one or many. And then we'll talk about the bond, or sometimes it'll be called a linkage, that is going to hold these macromolecules together. So the first category that we're going to talk about, that we'll probably spend the most time on, is proteins. So proteins, the monomer form is called an amino acid. The polymer is called a polypeptide. And the type of bond is called a peptide bond. So we'll talk a lot, probably the most, about proteins and spend a lot of time on all of this. The next are carbohydrates. So a lot of science words define what they are in the name. So carbo means carbon, hydrate means water. So a carbohydrate is made up of carbon, hydrogen, and oxygen. Water is made up of hydrogen and oxygen. The monomer form of a carbohydrate is called a monosaccharide. The polymer form is called a polysaccharide. And the bond that holds the polysaccharide together is called a glycosidic bond. And these are often called glycosidic linkages, same thing. The next is a nucleic acid. The monomer form of a nucleic acid is called a nucleotide. The polymer is called a polynucleotide. And the bond is called a phosphodiester bond. Again, sometimes you'll see these called linkages. And then the last that doesn't really fit this table very well because it doesn't form a polymer are lipids. Lipids are fats. And there's lots of different monomer pieces of lipids, and it doesn't really form the same polymer every time like it does with proteins, carbohydrates, and nucleic acids. But the type of bond is an ester bond. And we'll talk about these. These are specific to the lipid. So we'll talk about glycolipids and phospholipids and fatty acids and triacylglycerols and all kinds of lipids specifically. One thing that I want you to know is going to be a common theme through this class is talking about covalent versus non-covalent interactions. So all of these bonds here, these are all covalent bonds. And then something like a hydrogen bond, this would be an example of a non-covalent interaction. So you're going to see this theme kind of come up again and again when we're talking about all kinds of different processes, are the relationships between covalent interactions and non-covalent interactions. So that's something for you to kind of keep an eye out for. So make sure you read chapter 1. Make sure you look at the PowerPoint slides for chapter 1. And we will talk about water next.